Imagine living in a world where humans had the technology to control the source code of life. Well, the truth is that this technology already exists, and what was once thought to be science fiction is now becoming our reality, full of opportunities and challenges, and it's all down to the evolutionary new technology known as CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and is a simple yet powerful tool for editing genes. It was discovered in 2012 by American scientist Jennifer Doudna and French scientist Emmanuel Charpentier. Gene or genome editing is a type of genetic engineering technology in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. Humans have been engineering life for thousands of years by means of selective breeding in plants and animals. However, scientists were not quite sure how this occurred until the discovery of DNA in 1896. After this, the first genetically modified food went on sale in 1994. The Flavor Saver tomato had a longer shelf life than regular tomatoes due to the addition of an extra gene that could deter the buildup of a rotting enzyme. Although humans have been engineering genes for years, until recently genetic editing was extremely expensive, complicated, and took a long time to achieve. The invention of CRISPR shrunk the cost of gene editing by 99%, decreased the time it took for experiments from a year to a few weeks, and now anybody with a lab can use this technology. Today, you can even buy DIY CRISPR kits online. So how does this technology work? To understand how CRISPR works, we must first understand our own genetic makeup. Every living organism is built up of cells. Inside these cells are coils of DNA. Genes are sections of the DNA molecule. Each gene is made up of a combination of A's, T's, C's, and G's. CRISPR technology is able to map the genetic combination within DNA, allowing scientists to gather information on the DNA's entire genetic blueprint. Furthermore, it offers the ability to edit live cells, switch certain genes on and off, and target and study particular DNA sequences. In a nutshell, scientists are able to use CRISPR to search for a specific DNA sequence in the gene. Once a gene is chosen to be edited, an enzyme called Cas9 is introduced, which acts as a snipping tool. It cuts through the section of DNA, allowing that gene to be removed or replaced with a new gene, altering the entire genomic makeup of the DNA. This alters the cell, which then alters the organism as a whole. So why is this so important? Genes influence everything about a living organism, from our height, our eye color, to the resistance against certain diseases. But sometimes genes can carry errors, which can create faulty genes, and faulty genes can cause illness. CRISPR is able to pinpoint which genes are faulty and target the specific sequence to replace a faulty gene with a healthy one, or to change a gene so it behaves differently. This means that by editing genes, we can make radical improvements to human health, treating diseases such as HIV, sickle cell disease, and even some cancers. It is also relatively safe, as cells can be removed from the body, edited outside where you don't have to worry about delivering it to an entire person, and after the editing is confirmed, replaced in the patient. Scientists are already experimenting with creating a gene that is resistant to the HIV virus, and are currently testing it on rats. Cancer occurs when malignant cells refuse to die and multiply while concealing itself from our immune system. With the use of CRISPR, scientists believe they could edit genes within our immune system to make them better at hunting cancer. Apart from the improvements to our health, CRISPR can also be used in plants to make crops more nutritious, disease-resistant, and able to grow in difficult conditions. CRISPR can be used in animals to help them fight disease. For example, experiments are being carried out on mosquitoes with a gene that could make them resistant to malaria. These types of gene editing are known as somatic gene editing, which means the effect that the gene altering has ends with the individual organism and is not passed on to future generations. There is another type of gene editing, known as germline editing, which is highly criticised and is in fact illegal in many parts of the world today. Germline engineering involves editing the genes in human reproductive cells or early human embryos, which will result in gradual and irreversible changes to the human gene pool. This means that genetic diseases could be eradicated forever. However, it also comes with many ethical considerations. For one thing, not everybody agrees on which genetic conditions need fixing. Many people who live with dwarfism would not consider it a disease. In China in 2018, the first genetically modified twin girls, Lulu and Nana, were born. The Chinese scientist He Jiankui was fined and sentenced to 3 years in prison after he was found guilty of genetically modifying human embryos. The father of the twins was HIV positive, and the parents feared that the disease would be passed on to any children that they had. So they agreed to take part in the experiment. And now, they have beautiful, healthy twin girls. So what's the issue? Well, if germline editing were to be accepted and widely used, what would stop parents from moving on from just health benefits? If you could make your child immune to HIV, why not also give them an enhanced metabolism? 20/20 vision? Tall, muscular, blue eyes, blonde hair? Would designer babies become the norm? The biggest fear is due to the fact that with germline editing, any changes to the genetic makeup will be passed down through generations forever, slowly modifying the entire human race. So what's next for CRISPR? Designer pets? Will we take DNA from immortal organisms, such as lobsters, and begin experimenting with the human aging process? Could we someday be able to slow down, stop, or reverse aging? Engineer humans for life in space or underwater? Without proper restrictions, germline editing could be used to create superhuman soldiers and then be used against us. Or will we merely turn ourselves into a world that rejects non-perfect humans? The technology is already here. It's now time to decide if we use it or not.